All right, with this you get to see some real rockin' PowerPoint skills. With these diagrams, probably if you have different colors, it would be helpful, not necessary, of course, but when I normally do this at school, I draw them all on the board and use different colors and all sorts of stuff, but you just see how you want to go through here. Alrighty, so this is very, very simple. Again, if you're doing your PhD in this, it's going to be a lot more complex, but I basically tried to make these diagrams as simple as possible, so it makes it a little bit easier for you to understand how these immunotherapies work. All right, so the first one we're going to do are the monoclonal antibodies. Remember, these are the passive forms of immunotherapy. So we have our cancer cells, and remember with our cancer cells, they're very, very sneaky, right? So they're going to have some normal body proteins, like just your normal cell proteins on the outside. Because remember, these are normal, these are cells of your body that have run amok, all right? For some reason, something is completely bizarre happening in them now. So we've got our cancer cell here, but then they have sometimes these little weird proteins on the outside. So we've got normal cell proteins and we've got these weird cancer proteins. But your T cells, which patrol around all the time looking for foreign substances, when they see this cancer cell, they're like, I don't really know what's happening. It looks like it's my normal cell. I see all these normal, part of my body cells, but what's this little weird thing on here? Is this part of my, what is this? I don't know what to do. So it doesn't attack it because it doesn't know what to do with it. So what we can do in the lab is we're going to take one of these weird proteins from the cancer cell here, and we're going to put it into a petri dish. Well, we're going to grow antibodies in a petri dish. So we're going to make antibodies to that weird cancer protein. So in our petri dish, we're going to be making antibodies. So these are antibodies. Now antibodies kind of look like a T structure kind of a thing, but and this part of the antibody is going to recognize a part of the antigen. So the antigen is the protein or whatever on the bad thing, and the antibody is what your body makes to recognize the antigen. So we're going to make antibodies in the lab in a petri dish that will recognize what this little weird protein looks like. Then before I get there, just remember these things, they're very, very expensive to develop. They take a long time to produce as well, and there are side effects to them. For some monoclonal antibody, each treatment is $10,000, and a lot of times people need like 8 treatments of them. So that's $80,000 for a lot of this treatment. So it is very, very expensive. So what we're going to do now is we've got our cancer cell, and we're going to inject those antibodies into the patient. So the patient is going to get an infusion of those antibodies into their body. Alrighty, this is my great PowerPoint showing a syringe. That's supposed to be a syringe. This is supposed to be the needle going into your body. And those antibodies are now going to attach to those proteins on that cancer cell. So they're made to recognize those specific proteins on those cancer cells. And their job now is to say, hey, T cells, see this weird cell here that we're attached to? This is a bad cell. These are bad proteins on here. This is a bad cell. Come help and come and kill them. So the antibodies will, the T cells will attach to the antibodies and then eventually end up killing off those cancer cells. So that's how the monoclonal antibodies work. They're made specifically to recognize the odd proteins that are on cancer cells that your regular T cells can't recognize. This CA here, I should have said this earlier, this is just going to be short form for cancer. All right, so that's the first one. T cells come and kill the cancer cells. So our next ones are checkpoint inhibitors. So here's your T cells, and remember again, they've got the go switch or the go checkpoint, and they've got a stop checkpoint on them. So what normally happens is, in order for the T cell to turn on, there has to be something in there to turn that switch on, so something to activate it. So if there was an antibody that was in there, if it recognized an antibody, it would say, okay, well, this antibody is telling me that this is something bad, this is a virus or bacteria or foreign protein, something in there that's bad. So it will, the that antibody will activate that T cell. But what happens with cancer cells is they will activate the stop switch. So this is acting like this is your gas and this is your brake. And what happens is the cancer cell will fit in here and activate this and actually stop the T cell from doing its job. So the cancer cell comes in there, sits in there, and activates this activates this stop switch. So it turns that T cell off. So now the T cell doesn't actually go and kill the cancer cells because it's being turned off by the cancer cells themselves. Very, very smart. So that T cell is now turned off. So what we can do is, if we can somehow block this activation site in here so the cancer cell can't get in there and turn it off, and if we can, you know, like I said earlier, put some glue into the lock so that the little key can't fit in there, we can block the cancer cells from turning this thing off. All we're going to do is block it. We're not going to stimulate it or activate it. We're just going to block it. So what we can do now is we can inject these chemicals called checkpoint inhibitors, because they're going to inhibit the checkpoint into the body. And that means that checkpoint is blocked. So when that cancer cell tries to get in, watch this great graphics here. When the cancer cell tries to get in, it can't, it's blocked. So that way, when the antibodies notice the cancer cell, the T cell could actually go and do its job at killing the cancer cells. Now here's with your vaccines. We've talked about vaccines before. We've got a great big tumor of cancer cells. Now remember, with your tumors, with your cancer cells, they're not polite little cells anymore. They were regular cells, but they've gone crazy. They've run amok. Normal cells will be lined up nicely. They will stop multiplying when they get close to each other. They don't grow on top of each other. Cancer cells will overlap each other. They don't stop growing. They just continue to multiply over and over and over again. So that's what you end up with, a tumor. So what we're going to do basically is take one of the cells from the tumor. This is all going to happen in the lab. We're going to take a cell from the tumor and we're going to remove that from the tumor. Then from that particular cell, we're going to take one of those weird proteins from the outside of the cell. So we're going to take one of these things. Alrighty, so now we've got these proteins, but again, remember, your cells don't really know what they're looking for. They don't really understand that this is actually a bad thing that's in there, right? So they're going to take those proteins, put them in a petri dish, and then they're going to add an adjuvant to it, so a harmless protein, so that your body recognizes this as being something foreign. It's like it's like a little hand waver on the end there, or a little flag saying, hey, this guy here, he's bad, this is a bad thing. So we're going to have an adjuvant added onto that little protein to make it more recognizable. So now we're going to take that protein, all right, and we are going to, or not the proteins, we are going to take the proteins, sorry, I thought it was going over there, and we're going to inject those into the patient. So now we're injecting the proteins into the patient. And what's going to happen in the patient's body now, just like with a vaccine, the patient's body is going to go, whoa, wait a second here. What the heck are these? What is this? This little flag thing is telling me that this is actually a bad little protein in here and I shouldn't have it. So it decides to make antibodies to those odd proteins with a little adjuvant on them. All right, so now we've got our antibodies being made to these odd proteins because we've told our body this is something that's not correct. So now these antibodies can go around our human body, all right, and they are like, wait a second here, I know what I'm doing, let's go attack this cell, has these little weird things on here. Alrighty, so they're on the cancer cell now, and then they're like, hey, dude, T cells, get over here. We need you to come in here and do your job and kill these cancer cells. So the antibodies will kind of call the T cells and tell them to come on in here and get some work done. So that's how the vaccine works. Now again, you might say, well, why don't they use the vaccine instead of monoclonal antibodies? And the thing about cancers is they're very, very sneaky. Different cancers have different proteins on the outside. Different cancers, some of them are resistant to certain drug treatment as well. So you have to have a whole bunch of different ways in order to treat cancer. And sometimes with some of these treatments, they don't even work at all, even though they work theoretically, and they may work for your next door neighbor perfectly fine, but they may not work for you at all. Your body may, for some reason, just the type of cancer you have may just be resistant to them. A lot of times with the biologics, you can use them for a while, and then all of a sudden they don't work anymore. So they've got a whole bunch of different ways to do this, which is why it's so ridiculous when people say things like, CBD oil cures cancer, or, garlic cures cancer, because cancer cells are so sneaky and they have so many different ways that they outwit the body. So here's this is a boring one. Here's these boring, no, not boring, but the PowerPoint was kind of boring. These are the non-specific immunotherapy. So these are where we're going to take, or we're not going to take, but we're going to make the chemicals that normally would be in your body, and we're going to make more of them, and they're going to signal the immune response in your body. So we're going to produce proteins that are naturally made by the macrophages and the lymphocytes. We're going to make those in a lab, in a not really a petri dish, it's much bigger than petri dish, but this is just to show you it's in a lab. And what happens with these proteins is they only last long enough in the body to tell the immune cells what to do. You can't use these for long term because they do end up being quite toxic to your body. So we're going to inject those into the patient. Again, I got that again. They only last long enough to tell the immune cells what to do. So we're going to have a whole pile of these now, rather than the few little ones that your body are actually going to make. And they are going to increase. Some of the cells they increase are things like the natural killer cells. So these are a type of lymphocyte that we didn't really get into talking about. T helper cells, which is another type of lymphocyte, and dendritic cells. So we're going to increase the number of all these cells which are naturally occurring in your body. We're just going to make more of them so that they can go in there and fight. Now the last one is this CAR T cell therapy, this chimeric antigen receptor. I had a little bit of difficulty doing this and trying to make it really simple to understand. So I think this is good. So basically what we're going to do in this particular case is we're going to take the T cells from the patient this time. So we're going to take the T cells out of the patient. So the T cells, now remember with the T cells, I've just been making them just plain, but they do have proteins around the outside that are going to go around and recognize antibodies and other antigens or viruses or bacteria that are bad, all right? So they do have little receptors around the outside of them. I just haven't bothered putting them in because it just makes everything more complicated. But it's not just a baldy cell, they do have proteins around the outside. So what we're going to do is we're going to, in the lab, we're going to take the T cell from the patient, and in the lab, we're going to inject a very special type of virus. If you remember from the viruses, I had said that sometimes there are good uses for them used in biotechnology. So this is one of the reasons why they're used in biotechnology. So we're going to inject a virus into a T cell. Now that virus is going to have genetic material inside that codes for certain proteins that we're going to want to go onto the outside of the cell of our T cells, and those proteins that are on the outside are going to recognize cancer cells. So certain types of cancer cells, and this works very well for that acute lymphoblastic leukemia. So we're going to inject a virus into here that has the DNA to make proteins that are going to go on the outside of the T cell. Now it's called a chimeric cell because a chimera is an organism that has two distinct different types of DNA. So our T cell is going to have your regular cell DNA in it, and it's also going to have DNA from a virus that's going to code for some different proteins. So there's an episode, I think, on House where the person had two different types of DNA in there, and what had happened is the child or the person was a twin, and then sometimes with twins what happens is one twin engulfs the other twin, and then there's like the two DNA is mixed up into one organism. It's kind of neat, actually. If you look up chimeras, there's it comes out of mythology, but there are actual real animals that are chimeras as well. So basically now what's happening is that virus, the genetic materials in the virus causes the cell to make proteins which recognize the cancer proteins. So now what we're doing in here is that virus made proteins. So these little proteins that are in here are going to be ones that recognize the cancer cells with the type of cancer that we're talking about. So those proteins move and they embed themselves in the T cell membranes. So now this these membranes here, or these proteins here, are now embedded in that cell membrane, and these are specific for that type of cancer that we're going to be treating. So we're going to take these CAR T cells, because now these are CAR T cells. So they have their regular genetic information on the inside, and then they have new genetic information from another entity that is now embedded in their genetic information. So now the T cell can recognize the cancer cell. So I just made this one like this, little squares on the end, because the cancer cells, you know, have these weird proteins around the outside. So now that T cell goes, oh, wait a minute, I understand this thing, and will bind to that cancer cell and then be able to kill it. Right? So basically that's your diagrams for your immunotherapy. Again, in class, I think it would be a lot more dramatic and fun, and you know, chalk flying all over the place. There's a quiz on this, but this is going to be our last regular notes. I'm going to stop anything kind of new here, and we're going to have our last 2 weeks, we're going to kind of do something that's a little bit more fun and just a little bit easier for you guys, because I don't know how you've gotten through all this stuff, but I have admired your at how hard you've worked. All right, so have fun drawing this stuff out. Talk to you later.